All right, thanks. Thanks for the introduction, Erica. My name is Junsik, like RNA-seq Junsik, and Choi, in the Whitehead Institute, actually across the street. So today, I'm going to start with a generalized question first. So I'm studying plant biology, and you might ask, like, why do people study plant biology, right? Why do people study plants? First, they're actually very pretty, right? Second, they can be source of our daily product and it can be money, right? But for many plant biologists like myself, plants themselves are a great platform to unravel the mystery of organism and life. So the question now is like, which plant do I study? So let me put it in this way. When you want to have a pet, you don't really choose a giraffe, right? But you end up with usually a dog or even a cat or even a smaller one like a hamster. So this is quite similar to plant biologists in the reality. Not every plant biologist can study giant trees, nor they want to do that. So they can still study plants by choosing a smaller organism, such as Arabidopsis thaliana, that I'm going to tell you. So here is the Arabidopsis thaliana. They can grow up to like a 1 foot tall, so it's easy to handle and easy to grow in a lab condition like this. There are many different subfields of the plant biology, and I specifically study plant cells in Arabidopsis thaliana. So let's go look into a cell. So the previous speaker already talked about cell. This is a cell. I mean, it's generalized diagram, can be from like animal or either plant. And you already see there are many different subjects you can study even in a single cell. So I have to specifically choose which one to study further inside this cell, and I chose an organelle called nucleus, which contains genetic information or DNA. So again, we're going into a nucleus, and again, there are too many components you can study for your whole life. So today I'm going to just introduce one important structure, which is called the nuclear lamina under here. So what is nuclear lamina and why is it important? Why do I study that? So first, the nuclear lamina as a definition is a meshwork protein structure under the inner nuclear membrane. So you see that it's like a meshwork structure under this inner nuclear membrane. And because it plasters the inner nuclear membrane, it is in close proximity to genetic information or DNA and also other protein components. The other thing is that the nuclear lamina affects how cellular materials are going in and out of nucleus by interacting with a structure called nuclear pore complex. All right. The most important part of the nuclear lamina that I'm going to tell you is that if the organism have a nuclear lamina defects, then the organism will actually suffer genetic disease and abnormal development. So in plant biology that I'm studying, there are many different features of development or features of the nuclear lamina. And then one thing that I found is that they are very important in plant reproduction. So you're looking at here a Arabidopsis pollen. So this is a single pollen, and in a healthy, normal Arabidopsis plant, you can see this sleek pollen, but you have a defects in the nuclear lamina structure, then the pollen actually become oddly shaped in the mutant. The nuclear lamina itself is a small, tiny structure in a single nucleus in the cell, but the failure of having this structure will result into a butterfly effect, ranging from this reduced fertility to the complete death of the organism. So my main question, so by studying that, I'm sorry, this is because the mutated nuclear lamina cannot regulate genetic information properly and the nuclear function too. So by studying the plant nuclear lamina, I can illuminate the mechanism how the plant nuclei regulate the plant genetic information and plant genetic disease and overall plant development. With that, I really appreciate nanotechnology materials core facility. They really pushed me to submit this image. Without their help, I couldn't get selected here. I really appreciate Abigail and David for taking these images. And of course, this whole project couldn't be progressed with my great mentor in the Whitehead Institute, Mary Gehring. Thanks for listening.